Good morning, good afternoon, and good evening. Welcome to Farcast Biosciences Time Series webinar. Farcast is a leader in the field of human tumor microenvironment research, and our journey started at an MIT lab over a decade ago. Over the course of our lifespan, we have processed over 22,000 samples across a variety of tumor types. Farcast Time is a platform that has evolved to preserve human tumors in their native state. And as we will learn today, this technology enables an elegant interrogation of tumors by a variety of therapeutic agents and offers a multi-dimensional view of the impact of modulation on tumor as well as immune cells. This webinar is being recorded, and you are encouraged to send in your questions through the chat box. Your questions will be addressed at the end of the webinar. I would now like to introduce Dr. Nandini Pal Basak, Director R&D at Farcast. Nandini received her PhD in Life Sciences and Biotechnology in the area of cellular metabolism and is passionate about establishing a robust tumor microenvironment platform. She has made a significant contribution in the development of Farcast proprietary technology. And without further ado, let's welcome Dr. Nandini Pal Basak.
Hello, everyone. Thank you once again for joining in the first of Farcast Time webinar series. Today, I'm going to speak about human tumor model to study tumoricidal and immunomodulatory effect in solid tumors. In this webinar, I'm going to talk about and introduce you to the Farcast Time culture platform. We will see how we can evaluate the tumoricidal effect in Farcast time. And then we will try to understand one of the major component of the tumor microenvironment and how it responds in the Farcast time culture platform. At the end, I will answer to some of your queries. Please continue to write your queries in the chat box during the entire webinar. So what is Farcast time? Time, the acronym stands for Tumor Immune Microenvironment. So there are various kinds of model which has been used to understand the tumor biology. To understand the drug efficacy, the drugs which are designed to target the tumor cells, to study their efficacy, various kinds of model has been used. Classically, the 2D culture system was used, which has the advantage of high throughput screening, in which you could understand the molecular mechanism of the drug and see how it can affect by killing the tumor. Then we have the mice model, which gave a more systemic view of how the drug would work. With the new 3D culture system like the spheroids and the organoids, it also has the ability, or rather it has improved what we could see in 2D culture system by giving it the 3D contexture. You can do high throughput screening and do long-term culture in it. But none of these model system is able to provide a near native human tumor microenvironment representation. Now, why is it important to have a system or a model system where you can study the near native human tumor microenvironment? It is because there are certain drugs which has shown that the tumor microenvironment has the potential to regulate how it will function or what the drug's efficacy will be in that tumor microenvironment. So there is the histoculture concept or the model system which has the potential to preserve this native architecture and give a complete composition of this complex microenvironment. And Farcast time is such a histoculture platform in which we have observed that it can preserve the tumor immune microenvironment. So therefore, Farcast time, the histoculture model system, and it has this unique capability to help in various biopharma application to understand the way the drug is working or the drug's mechanism of action or efficacy. So I will briefly walk you through the workflow. So we acquire tumor samples from consented patients. Our specimens are surgical excess of the tumor tissue. We also have the matched blood of the patient. Now, once it comes to our facility, using our proprietary method, we process the tumor tissue such that it preserves the tumor microenvironment and we make small fragments or explants out of it. With the blood, we isolate the autologous plasma, which is a part of our culture system. Together, this is able to preserve the tumor microenvironment and keep it functional, and we can culture it over a short period of time. And during this culture period, we can interrogate it with various kinds of drug, their combination, or different kinds of concentrations. At the end, using different kinds of assays, whether it is an endpoint assay or a kinetic assay. So endpoint assays like IHC, flow cytometry, nanostring based assays, or kinetic assays like cytokine profiling or metabolic viability assays. So using all these different kind of assays, we are able to generate a multi-dimensional readout. And this kind of studies are done in a small cohort, say up to 10 to 20 patient samples. And through that, we are able to understand or get a deeper insight of what is happening in a cohort of sample. So what you can see over here is a schematic representation of the tumor microenvironment containing the tumor cells, the stromal components, the various kinds of immune cells. Now, to ensure that all this complexity is represented, one needs to have a fine balance between the optimal size and thickness. Because one hand you want to have a better representation of the tumor microenvironment. On the other hand, you have to maintain the size and thickness such that there is penetration of drug, there is penetration of nutrition or diffusion of nutrition through the explant. Plus the cytokines or the other factors which are secreted by the tumor cells or the stromal fibroblast or the various immune cells can come out of the explant and come in this culture supernatant in which we can study them. So in our platform, we have been able to find that optimal size and thickness which can maintain this fine balance. We do not use any enzymatic digestion. Now, the benefit of this is that the inherent stromal component or the architecture is therefore retained. And through that, we are able to achieve the preservation of the original tumor microenvironment. Now, this entire culture platform is compatible with any kind of drugs you want to test it with, especially with various kind of immuno-oncology drugs like the checkpoint inhibitors or the bispecific drugs, or you can do cytokine-based therapy, or even with oncolytic viruses. Then, of course, there is chemotherapies or targeted therapies which you can test, or even their combinations. Now, every drug has a different mechanism of action, or the way you would like to read out might be different. And therefore, what our platform provides is a lot of flexibility in how you can design your study, what kind of assay do you choose to understand the readout of the drug which you're working in, which therefore makes the study plan or the study design very flexible. In short, using our proprietary method of tissue processing, how we are making the different arms to treat it with drug or different kind of drug or their concentration, we are able to preserve the tumor microenvironment throughout this culture time. So here I just want to share with you a representation of how we are able to preserve this tumor microenvironment. So this is from a head and neck sample, and what you can see is hematoxylin and eosin stained sections. This is pre-culture, so you can see the tumor cells and the TILs in the stromal portion of this particular section. This is post-culture, where we have retained the tumor cells along with the immune cells in the stromal compartment, thereby showing that Farcast time is able to preserve the tumor immune microenvironment. Now let's try to understand in this platform how we can study tumoricidal effect. So to study the tumoricidal effect, this is our study design, where we have used head and neck cancer surgical excess samples along with matched blood, which will provide the autologous plasma. To observe the tumoricidal effect, we have used a standard of care drug combination, that is docetaxel, carboplatin, and 5-fluorouracil. This we have cultured or treated over 72 hours. In the study, therefore, we had a vehicle arm and a TPF arm. For the readouts, we used the hematoxylin and eosin staining, that is H&E staining, to determine the tumor content. We studied apoptosis by using IHC-based cleaved caspase tree, which was evaluated in the tumor compartment by our pathologists. Then we did an overall viability assay of the explant using the Prestoglu viability assay. So first I will walk you through one of the representative sample. So this is a sample which we have received. This is from a head and neck cancer patient, the tumor site being buccal mucosa. It was stage 1, grade 2 sample. This sample was processed and made into explants and distributed in 2 arms, the vehicle and the TPF arm. Now, pre-culture, we determined the Prestoglu readout, that is the baseline viability of the explants. Then it was treated either with vehicle or TPF. And after 72 hours of culture, we again did a viability assay to understand the effect of TPF on the explants. For this sample, what we found that in the TPF arm, there was a loss of viability of the explant. But is the loss of viability specific to the tumor cells? For that, we used the cleaved caspase tree expression, which was evaluated in the tumor cell compartment to understand whether there is tumor cell specific killing happening. So as you can observe in the top panel with the vehicle, there is hardly any expression of cleaved caspase tree in this particular section, whereas with the TPF treatment, we start observing tumor specific expression of cleaved caspase tree. Not only that, what we also observed that using the H&E sections, H&E stain section, that there is a change in tumor morphology and we observed a certain loss of cohesiveness. The cleaved caspase tree expression, when we quantitated, we observed that upon TPF treatment, there was an increase in the cleaved caspase tree expression in this particular sample. This study was done in 10 patient samples. So first, in this cohort, what we observed was upon TPF treatment, there was an overall reduction in viability as determined by the Prestoglu assay. We also observed a significant increase in the cleaved caspase tree expression upon TPF treatment, taking the cohort as a whole. In addition to this, we also saw a small change or a small decrease in the tumor content as determined using the H&E-based tumor evaluation. Now, each and every tumor sample is unique, and what we observe that this uniqueness of every tumor sample is also preserved in our platform by seeing that the effect of this drug is different for different tumor samples. So over here, as you can see, the fold change of cleaved caspase tree with respect to vehicle. So in this waterfall graph, what you observe is that each and every tumor sample has a different rate of response towards the TPF drug, as determined by the differential expression of the cleaved caspase tree. Now moving on to another important component of the tumor microenvironment, that is the immune cells. Now this immune cells is very important because all the current immuno-oncology based drugs are targeting this population either to reinvigorate it or to remove some suppressor phenotype. In our platform, we have observed that we are able to preserve the immune compartment. Here I will be talking about two specific compartments, the T cells and the macrophages. These two are one of the most important targets of various upcoming and ongoing research in immuno-oncology. In T cells are targeted to either activate or reinvigorate this population or to remove the suppressor T regulatory population. With macrophage, they are targeted either to change their polarization such that the tumor microenvironment can become more inflammatory in nature. So what you observe over here is the flow cytometry data generated from a sample which is cultured for 72 hours. And what you can see that we are able to retain viable T cell as determined by the CD3 expression and the macrophage population as determined by the CD68 positive population. Now the question is, these viable immune population which are present in our explants during the culture time, are they functional? Are they responsive to various stimuli? To test that, we have used the tumor explants and treated them with different activation molecules like the anti-CD3 plus IL-2 to specifically activate the T cell population and the macrophage stimulation agent LPS. To study the response, we used the cytokine release assay where we have collected the supernatant either pre-treatment, which we call as T0, or every 24 hours post-treatment, that is T24, T48 and T72. And using this particular cytokine panel, which comprises of some T cell specific molecules which are released upon T cell activation and some cytokines which are related to the macrophage activation. We also use the flow cytometry assay at the endpoint where we have dissociated the explants into single cells and then run this particular flow panel to understand the T cell activation status. So first we will take a look at the cytokines. We start with the T cell specific cytokines, the granzyme B, perforin and IFN gamma. What you can observe over here is that with the anti-CD3 plus IL-2 treatment, which is used to activate the T cells, we see a steady release of these cytokines over time. Such a release pattern is not observed with LPS treatment. Similarly, when we evaluate the macrophage stimulated specific cytokines, that is TNF-alpha, IL-6, IL-10 or IL-8, we again see a very specific response only from the LPS treated explants and not from the anti-CD3 plus IL-2. In the similar experiment where we have determined the cell level activity using flow cytometry, we have observed that with anti-CD3 plus IL-2 treatment, there is an increase in proliferation both in the CD8 compartment and in the CD4 compartment. There is also an increase in the activation marker CD69 in the CD8 compartment and CD4 compartment. We also observed that the intracellular granzyme B expression increases in CD8 upon anti-CD3 plus IL-2 treatment. But none of this is observed upon LPS treatment. So what this shows that in the tumor explant, where you have a complex mixture of different kinds of cells, we are able to elicit cell-specific response from this tumor microenvironment. So to summarize, the Farcast Time platform provides you a near native platform that can preserve the tumor and functional immune components for a 72-hour culture period. In this platform, we are able to determine the tumoricidal effect using cleaved caspase-3 expression, which also helps us get a differential response from different patient samples. Using the immunomodulatory agent, what we have observed that we can elicit cell type specific response from the tumor microenvironment. Thus, the Farcast Time platform gives an unique way to assess the efficacy of chemotherapeutic drugs or immuno-oncology drugs. Thank you, and now I will start taking your questions. Okay, so we have a few questions over here. So what are the indications this platform is available for? So what we have studied the head and neck cancer system, and what we have found that this is not only an indication which you can test, but also this particular cancer type can act as a tumor model system, because it has a nice repertoire of various immune cells along with tumor. This particular indication has SOC treatments, chemotherapeutic treatments, targeted treatments, and even the immuno-oncology drugs which are FDA approved for this platform. So in short, this cancer type has the ability to provide you as a model system to test various mechanism of action, either be it an immuno-oncology drug or any other kind of drugs. And we are also testing other system which we will come up with shortly, that is the CABR, GI or CRC, where you might have specific indication related therapies. Then what is the size of the explant? So the explants which we use are sub 500 micron in size, which helps us to maintain that kind of balance. Do you include PBMC in the culture? So we do get matched blood, and therefore we have access to the PBMC. For our regular culture, inclusion of PBMC is not mandatory, but if any any drug requires it as a mechanism of action, we can definitely include it. I think we are okay. We have some time left. So, have you looked at other cell types like NK cells? Yes, we did look at NK cells, and we have some level of preservation with NK cells, especially with CD56 DIM population. Um, okay, can we use it for blood cells that are grown in suspension culture? Can we use Farcast time to study leukemia mimicking bone marrow microenvironment, or is it suitable for solid tissue cancer in particular? So currently this platform is a model system for solid tumors. Therefore, with the current version, we are not able to do, or I would say we have not tested with bone marrow microenvironment. We would I would not be able to comment on how it will be preserved, but definitely with solid tumors we have tested. We have tested with various solid tumors, and you can use it currently for head and neck samples, but we will, as I mentioned, we will start with the different cancer indication. Okay, there are a few more questions. Have you tested the effect of anti-PD-1? So we had used anti-PD-1 therapy and we have seen their effect, but currently we are doing a much more thorough study. So it's ongoing and we have some encouraging results from it, and hopefully soon we'll be able to compile and present it to you. Okay, I think I have run out of time, but there are a few more questions. We will get back to you with the answers of those. And if you have any further question, or you would like to know more about our platform, or talk to us in a one-on-one basis to discuss your maybe any molecules which you feel that requires a human native tumor microenvironment system, please do write to us at biopharma@farcastbio.com. Thank you for being with us here today. And that's it from our end. Thank you. Have a great day.